Hello, my name's Christopher Bravery. I'm a consulting regulatory scientist at Advbiols. And today I'm going to talk to you about process performance qualification, or process validation would be another way of describing it. I'm going to talk mostly to the FDA guidance, primarily because I think it's more comprehensive and more usefully written perhaps than the European one. There is nothing contradictory about the FDA guidance versus the EU guidance. I know I've seen that some people feel that it's different. It's because there is more than one way you can undertake process performance qualification. And the FDA guidance really is about a life cycle approach. And I think this is a more practical way of doing things. But other variations are possible. And here I'm really going to focus on the principles of process validation rather than necessarily specific details about how to conduct it. One of the phrases I particularly like actually in the FDA guidance is to remind you that the perception of criticality is a continuum rather than a binary state. I think that's a very interesting way to look at it. So the question becomes, when do we start preparing for process validation? Well, clearly you're not going to do it in preclinical. You haven't even got a GMP process necessarily until later. First in human and other exploratory studies that you might classify as phase 2 type studies. You will be evolving the process, developing it, maybe enlarging it where necessary, etc. But it's not the one you're going to take through to commercial. So clearly you need to get to the stage of having a commercial ready process. And that might be exactly the process you're taking to commercial, or it could be a process that's designed deliberately to be easily altered to be suited for commercial to minimize the comparability risk of making process changes at that time. Either way, if you do that approach and that you do change it after approval, then obviously you'll need to revalidate the process. So it's also pretty important to go into the commercial phase with the process that is as close to your commercial process as possible, because at this confirmatory phase, what you might call pivotal studies, some people might call them phase 3, except that sometimes you try and submit phase 2 data, etc. Then this is the study that's going to provide the evidence for efficacy. So any changes to the product, i.e. through changes to the process after you've confirmed efficacy, are really going to require absolutely full and good quality comparability data. So that adds some risks, which means really you need to be preparing for process qualification even before you start your pivotal studies, or at least to do it the least risky way. And really what you want to be sure is that process that you have in place for the commercial is the right process, because you're going to use that for the pivotal and therefore you need to validate that one. And you really, really don't want to be making changes during the pivotal, which means that you need to design that process probably during your sort of phase 2 period. And that design needs to take into account not just the process, but actually the facilities in where it's going to be conducted or used. So you may be moving your process to another site for the commercial phase, because it may not be practical to do that before. So you need to make sure that the process and the facilities are well matched, because they will affect each other, obviously. And indeed, when I reflect on my experience, I know that moving commercial processes from one facility to another and then tweaking various things to fit in with the process of the way that the new facility works does sometimes lead to comparability issues. So that means that your process characterization ideally would be complete before pivotal, and a process control strategy would be in place, because then you'd be ready to do the process qualification. It could well be that some of this bleeds into the pivotal phase, but that really is very much at your risk. So obviously you don't want to validate a commercial process. And anyway, I've said all that. That's just text for the same thing. So these are a few snippets from the FDA guidance. Early process design experiments do not need to be performed under GMP conditions. That's just to say that you can do a lot of this development work outside of GMP. And indeed, really, the validation is really just confirming everything that you think you've established. So those studies that you do outside of GMP, of course, you want to mimic everything as far as possible, otherwise they won't be representative. A lot of process characterization, depending on the scale of your process, it would be unwieldy to do at scale. Perhaps even for some autologous processes, you don't necessarily want to run lots and lots of full-scale batches. So it can be extremely useful and maybe even absolutely necessary to have scaled-down models, either of parts of the process or the whole process. But there is a big warning that, of course, those models must be representative of the full-scale process. So any scaling must be representative of that, and that isn't always easy to achieve. Where the model doesn't match reality, obviously you need to understand where it doesn't and factor that in. And you may need to do some limited studies at a bigger scale. There is a lot of work to do to characterize the process, confirm the control strategy, etc. And so obviously there are tools like design of experiments that can avoid you doing huge numbers of one factor at a time experiments. However, you know, for some unit operations, one factor at a time may be adequate. I've mentioned that you need to show that you need to consider the design of the process along with the design of the facility. And one of the things that it's worth remembering is that you will need to be able to show that not just the process is operating, but all the support structures for the process. So the materials in, the materials out, the cleaning and all that sort of stuff, all of that, and the QC, all of that is also operating at the scale you wish. And indeed, here's an example from the KIMRIAR FDA BLA documents, the publicly available ones, and a question they got during review. Each patient lot is a separate manufacturing operation with several manual manipulations and incubation steps over a span of whatever period of time. Have you performed capacity studies, actual or simulated, to determine the number of lots that the facility, equipment, QC, and personnel can handle and sustain per day? And I think that's an important factor to take into consideration, particularly in this case where it's autologous, because you would validate one unit being operated or a few, whereas you may end up with a room absolutely cram-packed full of different operations occurring at a larger scale. So let's move on to process characterization. I think my first key lesson to say is that there's this sort of odd, I've had many odd conversations about unit operations where people have sort of described a step in the process as a unit operation. So here I have a manufacturing process, starting material, purification, activation, etc. Each of these is just a stage of manufacturing. What you really need to do for characterization first is obviously to identify all the individual unit operations. And so in my activation step, I've indicated that you, going into that activation step, are purified T cells, then beads are added for a bead binding unit operation, and then activation medium is added for an activation unit operation, and then at the end, perhaps the cells are washed. This is all purely hypothetical. It's just to give a sense of what I mean. So it's really important to break down to the unit operations so that you understand them, because in addition to any in-process testing that you may require for that unit operation, there are a whole series of process parameters that need to be established. And if you don't break it down properly, you'll miss these. So in a very sort of schematic way, you've got your starting materials, various raw materials going in, you've got a process, you've got your drug substance and or drug product, and then you're going to characterize the process and the product extensively with lots of different tests that fully characterize it or characterize it sufficiently based on your knowledge. Some of the testing you do during the characterization phase that I crossed out here will be of absolutely no use at all, and you'll discard them. You probably would have filtered those out actually in a more research phase, but you may eventually conclude that certain tests are not particularly useful. And so now you have a set of tests that fully characterize everything. And this could be quite a large number of tests, and it's very much bigger than your release testing and any in-process testing, hopefully. Then as you begin to understand the product more, the mechanism of action, and so on, then you will select some of those methods for routine release and control of the process. And it's important to emphasize here that it is the control strategy is an important consideration when selecting which release tests are necessary. I'll touch on this again in another slide. Now that you've characterized the process quite well, now you need to understand the impact of those process parameters that you have. And these could be temperature, they could be concentration of reagents, they can be duration of a step, they can be mixing, they can be oxygenation. All of these things are parameters that impact the performance of the process. And you need an idea of which are the critical quality attributes. And that can be difficult to deduce, but you'll have to work on best guesses if you're not entirely sure yet. And what you want to do is basically you'll have to do some sort of risk assessment and guess which ones are the most critical parameters and which are the critical quality attributes. And then you need to do some experiments to understand how they impact each other in order to be confirmed that the ranges that you're going to set are controlling the process adequately. That might be require, or it might be useful to actually explore a range of some parameters to the point at which they fail, i.e. the process fails, in order to be certain that you know where they are, and that so you can set a safety margin below which it won't fail, and so on. So process characterization then needs to really think about the purpose of each unit operation, such that you are looking at the right things, so that the critical quality attributes of the particular unit operation, if you like, so that when you explore the process parameters, you can see whether they're being influenced adversely or whether they're being kept within acceptable boundaries. So let's go back to this idea of the step of activation. The sorts of things you want to understand as parameters for this step might include some of the following. So for the beads, you might want to work out what's the ratio of target cells to beads to optimize. You might find that it doesn't matter how many beads you add too much, it doesn't alter the activation, but what it will do is cause you problems with downstream clearance of those beads. So there may be considerations beyond the particular unit operation. Now, there's an activation step here, and that activation step in this particular example is meant to, allow for the cells to be transduced by a vector. This is a CAR-T type example. So how do you know if the cells are sufficiently activated so that you can set, say, the duration and concentration of various things, or any, mixing or whatever? So one way might be, so transduction efficiency at the later step is an indirect measure of this, but you may want to look for a marker of the cells to see how well activated they are. This isn't something that you would necessarily have to use routinely, depends if you think that it's necessary, But it's a way of, in the characterization step, understanding how you've optimized the activation state of the cells. And these sort of tests would be used for enhanced testing during the performance validation, the performance process performance qualification, the validation. So these aren't wasted. This isn't wasted characterization at all. These methods will be used. Those same methods may well be extremely useful for comparability later as well. So slowly you chip away at your knowledge so that you can begin to build a proper specification, and you can set the process parameters such that you know they are controlling the process. Because when you test that drug product at the end, you're not controlling anything, you're just checking everything worked okay. The process control comes through the process parameters. And these are very often overlooked in documentation early in development. And I find it quite strange that the regulators don't ask more often, to be honest, for more detail. And eventually we come up with a comprehensive specification with lots of ins and outs, lots of maximums and minimums and so on. So having explored the impact of the process parameters on the critical quality attributes in a risk-based approach, almost certainly because of the number of possibilities, you'll now be able to set normal operating ranges if you like. But you may also know, you'll also have a characterization range, which is how much you've characterized, and you'll know, where the optimal set points are, etc., etc. And to come back to the overall setting of specifications for a controlled process and for release of the product. This is a figure that it comes out of a publication originally. Then it was modified a little bit by someone for an ICH Q5E presentation, which is where I noticed it, and I've adapted it further because I like the analogy. The analogy is to make the point that release testing is just the tip of the iceberg, yeah? So it is the, it confirms that a well-controlled process was effective, because what it doesn't achieve is complete characterization. That's under the heading extended characterization. These are all the methods that were completely characterized. And why doesn't it completely characterize the process? Because you have the process under control. So there's a limit to how far outside of your intended specifications the product should be able to be. If the process is fully controlled, then this subset of all the tests that are required for characterization should be sufficient to release the product. And that is an important consideration when you set that specification. And indeed, all the in-process specifications come out of all this characterization and demonstration that the process is under control. And the validation of your process aims to prove this. So there's the set of routine tests, and then you would want to do some enhanced testing during process validation runs. So there's some additional tests. And let's look at, go back to the FDA guidance, because they make a very important point that ideally you want to run everything at the set points. What you don't want is a whole series of optional runs that, test the extremes in different places. That's far better done in small scale models. What you're really trying to show is if you run the process under normal conditions, will it be well controlled? And because your release testing is a subset of all characterization, you need to enhance that release testing for process validation to confirm that your assumptions about the way that the process was controlled are correct. So you're generally aiming for midpoints. However, you should include really critical things like the starting material. If you're using cell banks, if you're using cell banks, I'm going to leave this on the screen so you can read it a bit, and I'll just pick a couple of points out. If you're using cell banks, then you would want to use some different donors. If it's an allogeneic product, then you definitely want different donors with different characteristics. And here is an example of some of the FDA's comments on that for Kymriah, and how they didn't really cover the range of variation in the starting material. Similar sorts of arguments from Yaskarta. I apologize for whizzing through these fairly quickly. And again, some criticism about ranges and things being quite tight and data not necessarily being shared. And if you get your process control strategy wrong or inadequately controlled, then of course you may suffer some of the issues of manufacturing failures, etc., after approval, and it's much more problematic in the commercial phase. So yes, include some different donors. Look at almost everything else in small-scale studies to avoid the need to include them into process validation. That might include things like validating out the removal of impurities, for example, as well. You need to understand the critical materials attributes of the materials you use. Some of the materials you use are extremely complicated biological materials. So you'll need to look at different batches over a period of time and understand what matters and whether you need to modify how you add it depending on the supplier. My penultimate comment is just to warn you about a statement I found in the FDA guidance about platform knowledge. In my view, the industry is not at a point where we have platforms. You may have several CAR-Ts under development and think you have a platform, but I think until you've had multiple products approved on a platform, you can't really call it a platform. So the manufacture of monoclonal antibodies would be a platform for sure now for some companies, but please don't be mistaken into thinking that this means that you can just draw from your other experience with other products and assume that they're okay. And that's it. Thank you very much. And the questions will come at the interval. Many thanks.